Hey, Mr. P here. In this video, we're going to have a quick discussion about the principle of segregation and how segregation can help us describe what's happening in single gene traits and how those single gene traits are inherited. In the last video, if you recall, we talked about how genes are passed on. We talked about why pea plants. We've talked about why Mendel chose pea plants. We talked about a little bit of crossing. Or a little bit about crossing in terms of pea plant traits and how they're inherited. And I left you with a question at the end of that last video where why doesn't the number of alleles and why doesn't the number of genes keep doubling in subsequent generations if we know that organisms have 2 alleles per trait or 2 alleles per gene, and we know that genes are inherited? The question was, why are the number of alleles not doubling every time parent plants, in this case, pass their alleles on to their offspring? And the question, or the answer that I left it with, was segregation. There must be only one allele passed on per gene and per trait to each organism, each offspring, in each cross. And so now we're going to talk a little bit about why that's the case, how we know that's the case, and what it looks like. Okay, so if members of each generation carry 2 genes for a given trait, there must be some way to prevent gene numbers from doubling in each succeeding generation. That was basically a question I left it with. Now there's a comment, or this is a particular comment that we will continue to discuss and elaborate on in this particular video. And so Mendel proposed that members of a gene pair must separate or segregate from each other during gamete formation. What does segregation mean? I know that that's a vocab term for this particular chapter, but segregation means that when an organism, in this case, is big R, or little r, they must separate their alleles and pass them on equally during gamete formation. So these organisms, this purebred round seed pea plant is homozygous dominant. This purebred wrinkled seed plant is purebred or homozygous recessive. They both or each have 2 alleles for that particular trait, that particular gene. And because they're homozygous, it means that when they form gametes, they separate out their alleles, and that each allele is equally likely to be passed on. And so this big R is going to be passed on half the time, and that allele is going to be passed on half the time. This allele will be passed on half the time, and this allele will be passed on half the time. Okay? This phenomenon is called segregation. As a result, each gamete receives only one of the 2 genes that control a particular trait. You can see that this, if this was our male, let's assume, okay, we can go over here and we can see that this is a male and this is a female. We know that sexual reproduction requires the crossing of a male and female pea plant, or a male and female organism of whatever cross we're looking at. Let's go ahead and assume or assign gender. This we'll say is the male. This we'll say is the female. These gametes are the egg cells. These gametes are the sperm cells. Okay? Eggs, sperm cells. And then when we cross these particular organisms, the organisms in our first generation, or F1, that are all heterozygous, because all of these organisms, 100% of our F1 offspring in this particular cross will be heterozygous. And so when we cross 2 heterozygotes, heterozygous, a male heterozygote and a female heterozygote, we will get the 1 to 2 to 1 offspring ratio in our second generation, or the F2. Okay? Again, just like in this first generation, these particular parents are going to segregate their alleles. They're going to pass this allele on half the time, this allele half the time. These parents that are now here will pass their alleles on half the time. So half of this male's offspring will get a big R from it, okay, these two, and half of its offspring will receive its little r from him. This female, half of her offspring will receive the big R from her, and half of her offspring will receive the little r from her. And when you put them together in a Punnett square, the big R come together and produces a homozygous dominant F2 offspring. Big R, little r comes together to produce a heterozygous F2 offspring. Big R, little r comes together to produce a heterozygous F2 offspring. And then little r is going to come together to produce the homozygous recessive F2 offspring. When you put them together, you get big R, big little, little in a 1/4, 1/2, 1/4 ratio, which is a 1 to 2 to 1. You can see that here. Genotype, 1 big R, 2 big R, 1 little. That's a 1 to 2 to 1 genotypic ratio. And phenotypically, because 3 or 3/4, 3 out of 4, all have at least one big R, they all will be round, which is the dominant trait in this particular cross, and 1/4 will be wrinkled. And so we get a 3 to 1 round to wrinkled because these two genotypes come together to produce the 3 to 1 phenotypic ratio, which is 3 round to 1 wrinkled. Okay? So, what is segregation? If we talk about it in terms of vocabulary definition, it is the separation of members of a gene pair from each other during gamete formation. Again, the segregation happens here during gamete formation. Okay, and there are gametes that are made here because our F1 plants need to produce gametes in order to produce F2. Our F2 offspring will need to produce gametes in order to make our F3. And so each offspring is going to be producing gametes via segregation, but segregation always refers to how alleles separate during gamete formation. Okay? Bring your questions to class. See ya.